Cancer detected.
He's ready for you, sir.
Cancer detected.
Took y'all a while to find the tumor, huh? Traitor! You're a tumor suppressor gene. You were supposed to help us prevent the cancer, not aid it. I have a different purpose now. I can't believe this. You know what, P53? Let me tell you a story. A story about a superhero who the whole body rallied behind. The guardian of the genome. All somatic cells go through 4 stages of life called the cell cycle. The G1 phase where they increase in size, the S phase where they replicate their DNA, the G2 phase where the cell continues to grow and prepares for division, and finally the mitotic phase, when the cell stops growing and divides into 2 daughter cells. Genes are specific sequences of DNA that code for a certain product, either a protein or RNA. Though rare, mutations are when the sequence of DNA is altered. An astonishing 120,000 mistakes are made every time DNA is replicated, but fortunately most of these are corrected by DNA polymerase. The rest of the errors are handled by the p53 gene. Based on the instructions from the p53 gene, p53 proteins are synthesized. The p53 protein is a transcription factor, which means that it helps turn specific genes on or off by binding to nearby DNA called the promoter. The first thing that p53 proteins do when DNA damage is found is activate the p21, GAD45, and 1433 sigma genes. The product of the p21 gene can arrest the cell cycle at G1 by inhibiting CDK2, a protein required for the progression into the S phase. The products of all 3 aforementioned genes can arrest the cell cycle at G2 by inhibiting CDC2, which is a protein required for the progression of the cell cycle into the M phase. By stopping the cell cycle, p53 gives the cell additional time to fix DNA. The p53 proteins then interact with and activate the p53R2 gene, AP endonuclease, and DNA polymerase II, all of which are heavily involved in the repair of DNA. The best part about p53 though? He was willing to sacrifice the whole cell for the betterment of the body if the DNA was damaged beyond repair. In this case, the p53 proteins turn on genes called BAX and APAF1. The products of these genes would then lead to the stimulation of me, caspase-9, and I would then initiate apoptosis or cell suicide at your command. Thanks to p53, the cell's DNA damage didn't lead to it dividing uncontrollably and becoming a tumor. The end. But that's an old tale now. The guardian of the genome has now led to the destruction of the genome. Wait! I can't control it. My DNA sequence has been mutated, caspase. How? First, it could have been an inherited mutation called the Li-Fraumeni syndrome. Or it could have been an acquired mutation due to a carcinogen. Regardless, once mutated, I lost my tumor-suppressing powers and gained functions that helped the cancer out. There's nothing we can do about it now. But what if we could? Researchers are evaluating certain small molecules and dietary changes in clinical trials for people with blood-related cancers that could reduce the inhibition of p53 by a protein called MDM2, which could help activate inactivated p53 genes. Who knows? Maybe the p53 gene will help mark the end of humanity's battle against cancer for good.